either the summary statistics or the HTC inventory. So we'll go into the HTC, we'll go to the donor list, and this is an important page, this overview page. So what I've added here, so these are all of the cases that are part of the inventory now. The DP are just those pilot studies that we did before we were really funded. But what I've added are these groups. So you can see as you float over these, you'll see known, no known, neonate, infant, child. You get the idea, right? So each one of those groups you can float over. And some of these first ones have been there for quite a while, but what I added was, for example, here, these 5, single-cell RNA-seq has been done at some point, either by us or by Cincinnati. And so those are the cases that have seen single-cell RNA-seq in that particular batch, that 2017 batch. The other thing you can do, each one of these groups, if you click on it, that'll give you just the list. So it stays there. You can copy it, whatever you want to do.
When I then go on that donor, it shows me what other samples you have of that?
Yes. Yep. I'll show you that in just a second, just to make sure everybody's oriented. This floats around a little odd. There we go. Okay, so what I added were a couple of the experimental groups, but also now some of the disease. So asthma, for example, if you want to know how many came in with a diagnosis of asthma, we have 7 donors now. You'll see a couple of those they came in status asthmaticus and actually died of their asthma, and some of them just have history of asthma. So then as you see there, whoop, mouse works kind of weird. There. So we have BPD. We have enough in what Gail has told me is saccular stage. I believe her, I don't believe me.
What does that mean exactly?
That in looking at, well,
Gail. You know, it's a spectrum, but in the morphogenic stage of morphology of lung development, human lung development, it's the stage before real well-formed alveoli.
It's an anomaly, basically.
No, it's not.
No, not necessarily.
Normal development.
But you're saying the structure of the tissue is as if it was
so in some cases, it is technically pulmonary hyperplasia because they have their term lungs that are immature for age, but often they're 32, 34 weekers where they should be in the saccular stages.
Okay.
Oh, I'm sorry.
Yeah, but so should they? Am I concerned that should they be considered normal? Because like.
I'll answer your question in a second.
Yeah,
I will. So there's also a category of normal growth and structure. Yep. Sorry.
Click to the left.
I'm trying. Oh. Okay. Okay. So that says 75, and I'll show you where you can bring up that list of 75, and you'll see that they may be normal growth in structure, but they may have other abnormalities. So you still have to be careful when you look at these groups if you're choosing cases. I'll show you that in a second too. Okay, abnormal inflammation. I think for some of our work, it is important to know if there's excessive amounts, if there's bronchopneumonia, if there's moderate or severe inflammation. You may or may not want to choose that case depending on what you're looking for.
Why do you have so many with inflammation?
Why?
Yeah. The large group.
Well, sorry. Yeah, 76. So these are, remember what we're doing here, right? So they're human cases that have come, that have died because of the severity of their disease. Very few of the older ones will have not been on a ventilator for some period of time. Ventilation, oxygen, induces inflammation. In some cases, there's evidence of aspiration, And so that might happen in the process even of a motor vehicle accident, et cetera. Severe trauma will trigger inflammation. So it's actually in some ways surprising that we don't see more. How much of it is infectious? So we're trying to figure out, we're starting to do swabs now of these tissues so that I'm not funded to do it, so when we're going to do it, I'm not sure, but we could do 16S, we could do microbiome studies. We have not yet started to sample different areas, but I'm kind of hoping in phase 2 that we can propose to do that. In some cases, virus has been detected, either clinically before the case came to us or that we've detected it. We have a panel of common viruses and infections that we have applied to some of these cases, so we know that they're true. So RSV, influenza, et cetera. Okay, and then there are a few pending histology. We get ahead of Gail sometimes. Not very often. And then bulk sort RNA, and this just reminds me to say, you see these two dates here, 8/25/16, so the 12 cases that you've seen data from now, both transcriptome, proteome, lipidome, we have just submitted another 14 for bulk sorted for RNA sequencing. So 2 years later, we're going to flesh that out, and I'll show you that in a minute. Sorry, I don't want to take so much time. Okay, so the other way that you can get at what's in the inventory fairly easily is to click on Overview again. Scroll down, please. Come on. Just delayed on the WebEx, although I can see it happening on my computer, but it's not. Look at my computer. There we go. Yeah, okay. Okay, so click on that if you saw that link. And this is the full data set. And so there is more information on some of these cases, many of these cases, but what I think is most important is here, for example, the pathologist overview. This mostly comes from right lower lobes. This tells you how many blocks, what blocks Dr. Deutsch looked at to come to this conclusion. There is also much more detail from her pathology review available if you want it. And then what I've done is kind of try to coalesce all the information into this column, this ClinPath diagnosis column. And so that's a summary of all of the other data. So, for example, you see this one, a child came in, had metapneumovirus and rhinovirus positive, but had normal structure and growth, and mild acute chronic inflammation. So for your study, you might be fine using that because you just are looking at structure, but if you're looking for inflammation, you may find more than, if you're not looking for inflammation. Right, you may find more than you want. Does that make sense? So, okay. And so the other thing I wanted to show you was if you go up to groups here, these are all the groups that were actually just on the left side before. So I'm going to go down to normal growth and structure, I guess, yeah. And so that 75 will be listed here, and so you can go through it if you like, and you see some just say normal structure and development, and so they really should be. Some have a little bit of inflammation, but they shouldn't have more than moderate. Some may have a little bit of autolysis, but if they have more than moderate, then it would not be in this category. One of the things I wanted to show you is it may come up here that some of these you'll find saccular as the description or cannulicular as the description. But if it's here under normal growth and structure, then it's normal for age. There are also some that are abnormal, right? They're not as far along in lung development as they should be for their gestational age. And so those wouldn't be under this normal structure and growth category. They would be, there's a category for delayed development. So the best way for you to figure this out is just go and play with it. I'll show you one more that people have been interested in, right, is the BPD, chronic lung disease. And this has kind of surprised me, actually, of how many we've accumulated. One of the other things to show you is if you sort by age, so ascending age, and then you sort by age units, that will put them in order. So this child had BPD, looks like BPD, born a little bit older than most of our BPD now, and then came to us at donation and death at 1.75 months. And then you can scroll your way through that, and you see that we have BPD from 2 months of age through 5 years.
Do you think have been exomed?
None. Exomed?
Exomed?
None.
Why not?
Well, I'm not funded to do it.
We've got a lot of other, I mean, you know.
Go ahead.
It's interesting. I mean, it depends on what you want to get out of it. I mean, there's a lot of other cohorts to do exome sequencing if you're interested in variation associated.
But it sounds like 20% of these would be candidate for finding something interesting by exome.
Well, I think the value would be the potential to integrate the genetic sequence data with the molecular data, but I think that's an open question about how valuable.
Yeah, we probably need a lot larger sample size.
Yeah.
Well, actually, you know all about that, so why don't you use that?
That's what you're trying to do. I mean, if you're just trying to get a couple of representative mutations that are associated with some little, like, hypo development, then...
Yeah, well, so I think, yeah, I don't know in the BPD that would be very helpful. I mean, most of these have a risk of being pretty preterm at delivery, and yes, it's true, why don't all 26-weekers have severe BPD, right? But there are some of the pulmonary hypoplasia or the cannulicular that's immature for age, there may be something there that we should...
survivor is actually more useful. Yeah, what's actually better is looking at extreme phenotypes. There is a 23-weeker who doesn't develop BPD versus a 28-weeker who does develop BPD because they are the kids who were both preterm at higher risk, but you have...
I would say, plus, you don't have a lot of clinical history on most of these patients.
We're working on it.
Makes it but it makes it more challenging for analysis. And some of the outliers, as you say, is the ones who present, idiopathically with pulmonary hypoplasia or present with VOD, veno-occlusive disease, in a neonate, which is extremely unusual. Those would be
So what I would want to do off of something like this, that where you can get after it by so many different levels of single cell analysis and validational molecular histology, prioritize, given all of this information, what would be a potentially interesting group to get genetics on, and then you can then make some secondary decisions about which ones where you could get a nice mix of genetics plus single cell going.
I think that's the sort of thing that we're hoping this repository will do, right? It'll allow investigators, yourself or someone else, to design studies like that and pull the cases that would be most useful to be done, whether it's us in LungMAP or whether it's...
I think that would be a useful paragraph for the grant.
Yes. But for the current...
the value of...
I could take a swing at trying to cluster out some of these guys, but any of you guys could do that, where you could just really highlight some individuals that probably have an underlying genetics and for some interesting reason, and then put it all out and see who would you, if you could only do 10, 20 exomes, who would you pick?
Yeah, I think for the LungMAP phase 2, I think that's on the boards, but I think for phase 1, I think the emphasis all on normal lung, and I don't think any of the research centers have actually done any preterm lung.
Not very much.
Yeah.
Some has been done, but not very much. And I'm getting requests now. Remember, this is open not only to LungMAP now, but to investigators. I'm starting to get requests for BPD or for other diseases. So I do think that those are, I agree with you. That wasn't what we set out to do in phase 1. There's still plenty to do in phase 1 for normal. Decisions will probably have to be made about what is really to be done in phase 2.
Yeah, one of the issues we have had in phase 1 is because in the mouse we start from an earlier I would say, morphological time point, that when you're still in the sacula. But for human, we are starting from birth, which is you're already having alveoli.
Yeah.
So we are starting at a later.
Yeah, so that's actually why I found it. I'm kind of pleased that we have some canulicular and saccular in there, because some of them are younger gestational age. I can show you that too, but some are abnormally immature. And why is that? What controls that?
Well, in the CTs or all of them?
Mm-hmm. You should have. Yeah.
So the data Lindsay just presented today, the two of the postnatal data and the human, one is premature.
Yeah.
We actually can see some of the signature shift, especially in the type 2. But we didn't go there because we only have N-equivalent for both of them. They still cross together, but you see a clear shift on the type 2 cells.
For the two subjects?
Yeah.
We'll take a look at that.
Tom's going to, if I give him enough time.
Maybe it's in the data I'll show.
Yeah.
Yeah, we discussed it in the lab meeting, but it's, we didn't emphasize because it's N equals to 1 for each, right? It's interesting, but it's,
We're here to generate enthusiasm and hypotheses, so I agree. Our N's are small so far, but...
So there are for the exon sequence project, and I actually talked to the PO who's running that program. The structure is that they have a sequencing facility in capacity, and people who want to do the sending an application mentioning or state scientific need, why we want to sequence this cohort. So I actually mentioned our LungMAP data to the PO who's running that. So he said, one is normal, they are interested in disease. Secondly, they think they're running a much bigger cohort. They think our sample size is too small.
Yeah, how many of their samples are going to be good for single-cell analysis and validation?
Yeah, so with that, maybe I think to insert into some of our best cases for sequencing, that's a possibility.
I mean, it's very high risk, but I think it may be worth trying proof of principle if we're collecting all this molecular data, that to have those additional data about sequence variation could potentially be informative.
Yeah, so
I would assume that you'd get around 25% yield out of it.
Oh, I think so. Maybe better. Maybe more.
But
I mean, I think actually, an N of a prime N of 5 or something would be fine to have a look, and you're going to find something.
So the good thing is that it's not going to
causal variance.
Well, that will then justify doing a larger cohort.
I don't.
Yeah, we just need to write an application to say why we need to do this, why there's a sense.
These kids are
You need severe cases.
They're the ones that have bad luck.
Significant developmental pathology.
I mean, we've done a lot of these. We've done a few hundred. So.
Yeah.
Not LungMAP.
With severe developmental pathology?
Not severe, but...
We don't, in most cases, we don't know their developmental pathology. We just know that they do or don't have clinical disease, right, for the ones that have been, the exons have been done. I'll just point out here, too, that these have not all died of their BPD, right? So we have some reparative, I can call it that, reparative BPD. So I don't think we've had much of an opportunity to really look at what happens to these lungs on a molecular basis as kids get older, even if they survive.
Right?
So have you
know what I mean?
Yeah, ATAC-seq would be really nice on that one, because you will get all the
ATAC-seq.
You get all the epigenetic changes that might actually manifest in resolving or not resolving BPD.
Yeah, true.
So have you distinguished between what used to be old BPD, in some cases we still see that, and the new BPD, or the more milder?
Yeah, good question. So I actually noticed that, so Gail is my go-to here, right? I have an idea of what they should look like, but Gail really does. So I noticed this 23-weeker, she has called new BPD. So
that means the lack of remodeling.
Yeah.
So, yeah. I do distinguish between the two, whether they have a lot of lobular remodeling, which is more in keeping with the old BPD versus what we call the new BPD with just the simple distribution.
Yeah, and this one particularly interesting, a 3-year-old, but was not the victim of the BPD, but was neurologic. BD is brain death, not BPD. Okay, I think that's all I wanted to show you there.
Thank you.
Sure. I'll go back and,
So it says on the website, it said like other diseases, and then there is other tabs for every disease or lung disease. Can they all kind of be under lung disease? And in every category, there is not in any category, yet they're under one disease category. So it's a bit confusing.
yeah, so we are the victim of the way this works.
Okay.
The software works to some extent. It's not perfect. So I'm sorry, what are you saying? Oh, back on the overview back here?
Yeah.
Yeah. So when it says not in any group, that means not in these three groups for age of birth. Okay, so not in any group. If I click on that one, it brings up 192 because there are 5 that are actually in that sub-study. See what I mean? So that not in any group just refers to that group.